Does that sound better?
Yeah.
Okay. All right, so jumping right back into this. Let's start talking about what's going to happen in each of these different stages of this particular process. So glycolysis, the citric acid cycle, and oxidative phosphorylation. Remember again, oxidative phosphorylation makes the bulk of the ATP that's utilized later on by the cell, while the citric acid cycle and glycolysis make smaller amounts due to the way in which they phosphorylate. So they perform what's known as substrate level phosphorylation. The way substrate level phosphorylation works is you have a substrate that has previously been phosphorylated. You're going to use that phosphate to phosphorylate a molecule of ATP, which one time you'll then have a product and a molecule of ATP. Seems simple enough. The problem is, most of the time, this substrate has to be phosphorylated by using ATP initially. So what we're going to see with glycolysis is that we actually have what's known as an energy investment phase that happens during glycolysis, where we actually have to use ATP before we can start making ATP. So this is substrate level phosphorylation, and it's only performed by glycolysis in the citric acid cycle, not oxidative phosphorylation. Okay, so as its name implies, and as we've discussed numerous times before this, glycolysis is the splitting of sugar. So what we're doing overall in this process, in glycolysis, what we're doing primarily is we're taking a molecule of glucose and we're splitting it into two molecules of pyruvate. Two identical molecules of pyruvate. Now how many carbons does glucose have? 6. Okay, so if we're making two pyruvate molecules, we're actually not losing any carbon during this process. How many carbons are in each of those pyruvates?
3.
3. Okay? So remember, keep in mind that we're making two pyruvate molecules. Now, what's going to actually have to happen is during glycolysis, we're going to actually need to invest energy before we can start producing energy. So we have an energy investment phase and we have an energy payoff phase. Word of warning. I'm about to show you a couple of slides that are going to be crazy in terms of the amount of content they have on. Deep breath. I will do my best to explain everything and also stress to you what is the most important aspect of this lecture or any of these parts. Okay? We're about to get kind of crazy, but just stay with me. Okay, so overarching view of the energy investment phase. What's basically happening in this phase is we're investing adenosine triphosphate. Okay, so we're investing energy and we're phosphorylating that glucose molecule. Okay, so we're going to phosphorylate this glucose molecule. And eventually, that glucose molecule will be split apart and the phosphates that are attached to it are going to be the phosphates we use to phosphorylate ADP to produce 4 molecules of adenosine triphosphate. So, please note, we use 2 molecules of ATP during glycolysis. We produce 4. So there are 4 total molecules of ATP produced, but only 2 molecules that are net produced during this process. Does everybody see that? We had to use 2 to make 4. We only technically made 2 new ATP molecules. The rest basically making up for what we had to invest. Yes?
So basically, the energy payoff phase is whenever we produce 4 ATP. That's like...
Yeah, exactly. So think of it just like investing money. The initial part is where we're investing the energy or the money into whatever it is, and then if you make money off of it, that's where you're getting paid off. Okay? So we invest 2 ATPs, we make 4 ATPs. So if you're using ATP, it's the energy investment phase. If you're making ATP, it's the energy payoff phase.
Just out of curiosity,
Yes, Richard.
half of the phosphorus that are coming from the what do you call it, the stratum, where are the other half coming from?
Where the other phosphates?
Yes.
So you're saying, okay, well, we just used 2 here, where the other 2 coming from?
Correct.
There are additional molecules that we're going to pull those phosphates off of. They're not ATPs, but they're other either carbon-containing, inorganic, or organic molecules that have those phosphates attached.
Okay.
Okay? And we will use an enzyme that will allow us to remove those phosphates. Go ahead.
So the question is, how many ATPs are needed during glycolysis in phase 2?
You have to look and read. If I ask you how many ATPs total are made, then it's 4. If I ask for net, then it's 2. Okay? I'll probably use gross or net, because gross means, you know, that number there that we made, whereas net means take this number and subtract that. Now, the other thing I need you to be aware of is the fact that, remember, while we're performing glycolysis, we're actually performing redox reactions, so reduction and oxidation reactions, which is Allison's favorite topic. While we're doing that, we're going to actually be removing electrons from some of the intermediates here and transferring those electrons onto these molecules of NAD+. Remember, NAD+ is an electron acceptor. So it accepts those electrons and becomes reduced to NADH. Okay? So we form NADH and we form protons during these redox reactions. I'm stressing that because, again, the NADH molecules and the FADH2 molecules that are formed during this process are going to be key when we get over to oxidative phosphorylation. Remember I told y'all last week I got really excited in a totally nerdy way about oxidative phosphorylation? It's because I think it is one of the coolest processes in the way that it works and how it actually benefits from some of what we might consider otherwise byproducts of the glycolysis reaction. Okay? So, again, 4 gross ATPs are formed, 2 net. 2 NADHs are formed through the redox reactions, and at the very end of all of this, we also form pyruvate and water. Questions, concerns? Yes, Allison.
So the NAD+ is the electron acceptor, and it's the one that's reducing.
NAD+ is the electron acceptor, which means it is being reduced. So it is the oxidizing agent because it's being reduced. NADH will later donate the electrons it's carrying, which will make it the reducing agent, but it will be oxidized back into NAD+. Okay. All right, so let's have some fun and start actually looking at the intermediates in this whole process. Now, a couple of things. First and foremost, typically I'm not going to sit there and ask you to memorize the name of every single one of these intermediates. I'm not going to ask you to recognize them by structure or anything like that so much, because I don't think that that's really what's the most important thing here. What we are going to talk about, though, is what's happening and the enzymes that are used in each and every one of the steps involved in this process. Okay? So first and foremost, we have glucose. We need to actually split glucose apart by the end of glycolysis. So we're going to do that by technically marking it with phosphates. Because by marking it with phosphates, certain enzymes are going to actually be able to recognize it when it is phosphorylated. And eventually, an enzyme is going to be able to recognize a double phosphorylated structure and split it in half. Because we're trying to get to 2 pyruvate molecules, which means we need to cleave this glucose and get 2 3-carbon-containing molecules. So the first enzyme that we use is called hexokinase. And I will tell you, and you need to know this even beyond this class. Anytime you see the word kinase, first off, we know it's an enzyme because it ends in ase. But kinases specifically are enzymes that transfer phosphate groups. So anytime I see the word kinase, I should immediately be thinking that I'm going to see something add a phosphate onto my molecule or transfer it off to another molecule. And that's exactly what's happening here. We've got this particular kinase called hexokinase. Why do you think it's called hexokinase?
There's 6 of them.
It's a 6-carbon sugar, right? Okay? So it's a 6-carbon sugar. It's an enzyme that transfers a phosphate to a 6-carbon sugar. So hexokinase results in a phosphorylation of glucose to where now we have glucose 6-phosphate. Called glucose 6-phosphate because that phosphate is attached to the 6 carbon. Bless you. So even if I were to actually ask you about this particular molecule, if you know what glucose looks like, it's not that difficult because the only difference between this molecule here and this molecule here is I've added a phosphate onto the 6 carbon. So we've got glucose 6-phosphate, phosphorylated, using hexokinase. Now, we're going to use a particular enzyme called phosphoglucoisomerase. It's called phosphoglucoisomerase. Let's think about why. First off, it recognizes a phosphorylated glucose, which is why it's called phosphogluco. Okay? What do you think an isomerase does?
Splits isomers.
It switches between isomers. You'll notice this is a 6-sided ring. Okay, it's a hexagon. This ring is now a pentagon. Okay? So what this has effectively done is it has switched it from glucose 6-phosphate to fructose 6-phosphate. So phosphoglucoisomerase recognizes a phosphorylated glucose and facilitates the alteration of that glucose 6-phosphate to fructose 6-phosphate. So it allows us to switch between isomers. Questions, concerns so far? Yes, Allison? It facilitates the alteration of glucose 6-phosphate to fructose 6-phosphate. We're switching between isomers here. These are still isomers. This is just glucose 6-phosphate. This is fructose 6-phosphate. Yes, Heather?
Does the 6 have to do with the number of carbons in the ring?
Whenever you're looking in organic chemistry and you see a number, it typically corresponds to that particular carbon. So again, if we look here, this carbon at this point here is 1, 2, 3, 4, 5, 6, and the phosphate's attached to that 6 carbon.
You always start numbering from the carbon closest to the oxygen molecule.
You usually look at functional groups in organic chemistry, and that will tell you how you're going to actually number your carbon. Okay? All right, now, just to break off for a second, all of you did the respiration lab last week, all of you did fermentation. Who here saw that their sucrose actually wound up having a higher level of carbon dioxide made in a shorter amount of time than their glucose? Okay, all 2 of you. Awesome. Okay, well, for those of you who did see, and for those of you who might have wished you had seen it, the reason why sucrose actually wound up producing more carbon dioxide in a shorter period of time is because when you hydrolyze sucrose, you produce glucose and fructose. The glucose starts glycolysis right here like it normally would, like in the glucose tube. But the fructose actually hexokinase results in the phosphorylation of fructose, and fructose immediately becomes fructose 6-phosphate.